Mm. Okay, it should be good now.
Mhm.
All right, welcome everyone for this new webinar by MoroccoAI. We have the pleasure today to host Dr. Chaimaa Tarzi. So Dr. Chaimaa Tarzi, let me read you her biography. She's a lecturer and early career researcher at Teesside University, whose research focuses on computational microbiology, genome scale metabolic modeling, and machine learning applied to the human microbiome. She develops community metabolic models and interpretable predictive methods to understand how microbial interaction shape disease-associated states and to generate biological interpretation. Thank you very much, Dr. Chaimaa Tarzi, for accepting our invitation. And yeah, we are very looking forward to your presentation titled Predicting Human Microbiome Behavior in Diseases via Pairwise, Metabolic Modeling and Machine Learning. Thank you very much, and I'll leave you the floor.
Yeah, thank you again, Edwan, for the invitation, and thank you everyone for joining, of course, today session of webinar. First of all, it's already been introduced. I'm Chaimaa Tarzi. I'm an early career researcher, and today I will take you through how we can use metabolic modeling and machine learning together to predict how the human microbe behave in disease and in a healthy behavior. So we can say the goal is to make this accessible to AI scientists and also to bridge the gap for like interdisciplinary people that also work in biology system. So first thing I will go through is the outline. So we'll start a little bit easier so we understand the concepts behind of the microbial. So I will try to make you understand how the human microbial are important, the challenge that we have using and trying to understand those microbial behavior. And we will move on to the introduction to the genome scale metabolic model. So here I will take you from the genome scale, from the sequence itself to the genome scale metabolic model. And so how we create this mathematical model. Then we will go to the third part of the talk where we will mainly understand how we jump. So now we have this mathematical model, how we jump now for a simple a mathematical model of the bacteria to the complexity of the pairwise metabolic model, how we create this communication between the two metabolic model. And finally, of course, I will show you a use case scenario on how we use this technique to predict the disease state in biology, and how we will gain some biological interpretation and insights. And finally, we will merge to how we can apply this in a broader, we can say, case of scenario, and how we can do to make it more complex and see how we can integrate it in a future direction. So first thing is we can say is the microbial community. So if I may say, when we talk about the microbial behavior, we need to understand that the human body. The human body hosts trillion of microbial. So here we are talking about microbial communities such as bacteria, fungi, viruses, and forming a sort of consortia of microbiome. So this helps digesting our food, also help us to have a better immune system, and even influencing like our mood and the cognition. So the things when we see the human body, we try to understand the complexity, but still it's hard to, we can say, replicate exactly what it's doing. So when we are talking about here the balance, here I'm trying to understand, like, when there is any interception or any problem that we can say interfere into that balance of the microbial communities, where we have some condition known as this biosis. So we often see links to obesity, diabetes, cancer, and all this is based on this, we can say, gut-brain axis. Okay, so now we understand a little bit more on the we can say the silhouette with the icon, and for the gut, the lung, and the skin microbiome, and how they are important for the human body. We need now to capture the challenge of this microbial interaction. So first thing is, when we talk about the challenges, I want to highlight here the complex network of microbial species that connect here all the network together. So I said, like, we have thousands of species interacting dynamically using the exchange reaction. So in this case scenario, I just put here some, we can say, an easy way to understand a two species interaction, but the complexity or the challenge that we might have is to take it and experiment the bacteria in a isolation state. So we cannot capture the scale of it. So we need the computational framework that bridge mainly the biology mechanism and the data-driven inference. So I mean, we need a perfect collaboration between the biology and AI to be able to understand the complexity of those microbial community network. So final stage I want to explain before we can jump to the genome scale metabolic model is the metabolism. Why we need to understand the metabolism. So there is some people that have a small awareness. There is ATP productions, there is the waste release, all those, we can say, pathway that will help you understand the cell diagram, show like nutrition intake and then the production. So what I mean by this is, like, mainly when we talk about metabolism, is a total of all chemical reaction that will operate in a living organism. So as I said, is the intake vs the production. So in the metabolism, we have like a different cells. So the nutrients come in, okay, and then the energy is built in block and can be produced, or there is also some waste expelled in the host. So all the mapping of all this will help us to understand the reaction and the metabolic enzyme that regulate those reactions. So here I just put together like the importance of the metabolic and how it's efficient in the production also of biological products. Okay, so when I talk about metabolism, I need to like now make this to let's say a better like network. So if we understand like the differences between the production or the process that come between the reactant and how the enzyme interfere to like emphasize the production of specific products, it's here where we can understand it as a mathematical model. So what I mean by this is, imagine you in a simple like case of scenario. So here I put like just a simple a unique cell where there is the cytosol, there is the extracellular environment, and then they have different reactions. So there is reaction 1 till reaction 7. Okay, so from reaction 5 to reaction 7 are the transport reactions. So those reactions are those one, we call them transport one or exchange one, because they interact with the host, or they interact with the other bacteria. It's like they are the call center. They are the one who take the phone and tell you, okay, we have some energy, let's take it in, let's produce like from it like the biomass. So these are the important reaction that occur. But if we want to understand this mathematically, we need to realize it in a better way. So if I may, introduce how we start from like we can say the genome scale metabolic model to the reconstruction of the model. So here I will take a little bit more time because it's here where I will mainly explain the mathematical model that will, we can say, we get as an output for our, we can say, machine learning pipeline. So first of all, we have this genome sequencing, okay? So in this genome sequencing, we have like different sequence, so your DNA that will identify each, we can say, bacteria, fungi, or a specific, we can say, component or species, okay? So starting from this, okay, we will use a tool that will map the sequence, do a sort of a multiple sequencing, okay, mapping, to understand if like we can get using BLAST or using other tools, like some mapping to those reactions. So each sequence is related to those reaction. Each reaction have specific metabolic to be consumed and produced, okay? So what we try to do, using this mapping of sequences, we try to reconstruct we can say a network of all the reaction and metabolites that are produced and consumed. But we can say the constraint that we need to put to the model is wherever this species consume need to be produced. So it should be a balance between the consumption and the release, okay? So next stage of the pipeline, it's we can need, when we reconstruct the model, some of the reaction have problem on, we can say, there is some gaps, there is sometimes it's infeasible. So we need to identify those metabolites and see, based on literature, if this reaction is well, we can say, mapped when we do the networking. So if one, we can say, metabolite is not produced, or there is an infeasibility, or there is, like, we can say, something produced, but we don't know from where. We need to understand this gap, and then we call it gap filling, because it's here when we try to make it more feasible, our mathematical model, where we gap fill those holes to make everything makes sense when we are running like the equation, the matrix that we want to define, and when we want to quantify this growth rate. So we map, we try to fill those gaps, we try to do the gap filling, and then we can move to like the final thing is here where we try to test our model. How we test the model? We test the model basically on running it on we can say a specific diet, and see the feasibility of the model. Is he able to like simulate and quantify the growth rate on a specific diet condition? So what I mean by this is, like, I'm having like a constraint. I'm having like, look, you have this type of, we can say, oxygen intake. You have this type of, like, let's say, glucose intake. And what all this are within, I input it in the model as a constraint. So I will not have an open, we can say, solution interval. So I make it a little bit like a cone. And then I quantify this, we can say, fluxes. And of course, when I do this, when I do the testing of the model, I don't look just on the feasibility of the model, but I look also on the annotation and all the things that make the model a high quality model, okay? So now we understand a little bit more from the genome perspective to the metabolic model perspective. So if I want to move on, so just a glance of the last parts that I explained is like how we really quantify these fluxes, how we can understand the final equation that we want to solve. So you remember I explained the reaction and the metabolites, okay? So imagine those reactions are in your S matrix. Those reactions are your, we can say, columns, okay? And then each metabolite is the row of the matrices, okay? So when I will go to the reaction one, in this case, what I have in the reaction one. So this triangle has been consumed. So the species eats this small triangle. Imagine this triangle is, let's say, the hydrogen, okay? Then also there is another, like, let's say, and it produced, like, for example, hydrogen, okay? So it's consumed the same species and then give us, like, let's say, the other species. But sometimes there is not just one metabolite involved. Sometimes if you see in the, like, let's say the reaction, let's say the reaction 5, it consumed, let's imagine, consumed oxygen and then consumed hydrogen and give us the water. So this is a simple chemical reaction. Its V1, V2, and V3 are those, we can say, fluxes that are related to each reaction. So what we want to quantify, this V1, V2, V3, so the flux vector. So we have the reaction, we have the metabolites, and then we try to solve an equation where SV is equal to 0. And then to make, again, the solution space more narrowed like a cone, we can try to add more constraints. Those constraints, where we will add them? Basically, we add them based on the environmental constraints, or we can add them from the like, we can say, the gene expression. So any other information that will give, like, an optimal, so help us to narrow down the solution space. So when we identify the optimal solution, it will be easy to quantify the growth rate. Okay, so far we went on the importance of the microbial. We understand a little bit the genome scale metabolic model, the reconstruction pipeline, and I made you understand the flux balance analysis because it's one of the important features for our machine learning pipeline, okay? So now we understand the single species metabolic model. We need to understand the complexity because in our human body, unfortunately, there isn't one species and we don't have interaction. The complexity of it is everyone interacts with everyone. So this is why we need to understand how they are interacting. So in this case of scenario, we have, like, different interaction scenarios. So either the species interact with the species, and this is, we can say, one small complexity. We put it a little bit in a small scale complexity because it's just like a pairwise interaction. Is it still good because it gives you an idea on how you simulate the two species at a time, and help you also to not have a complex scale, so it's easier for the machine learning pipeline. But there is also other case scenario where there is, like, the microbial host interaction model. So here all the we can say species are in one, we can say, model, and this mega model, this mega big model, imagine it is a big bag containing all the species, and this big bag interacts with the host. Or the final one, which is it's very beautiful because it's captured all the complexity of the community metabolic model, but it's in terms of scale, it's a little bit complex. So here they interact with each other and they interact with the host itself. Okay, so now we had all, we can say, the necessarily baggage. So we can take the flight to the main, we can say, framework. So here is, like, the papers that we did in transit in metabolism that capture and describe all the pipeline. Is a small user guide for those that want to integrate microbial community in their work, and these are small tips to how you can use it to build the community metabolic model. Okay, so now we can go to, as I said, the framework overview. So in the framework overview, so what I want to explain here is we start from two different cases. And in this case of scenario, we have COVID-19 because like in 2019, we were working as my PhD was funded by a company called Complement Genomic, and they were doing tests of COVID in the pandemic. Okay, so we tried to contact them to try to use like the sequencing that will help us understand the virus based on the bacterial behavior. Okay, so but you can still keep in mind that this model can be used for different case scenarios. So imagine any virus disease, how we can say the changes on the microbial consortia can influence having or not having the virus. Okay, so if we start with like here, the beginning, in this case of scenario, you have two, we can say, subjects, so a healthy subject and a COVID-19 subject. So what we did is basically we built the Pairwise genome scale metabolic model. So you imagine, so starting from the genome scale, so each patient, the complexity of it, each patient have different bacterial types. So some people have some bacteria that are within their gut, some others disappeared, some of them like are there, but like the abundance, the relative abundance are lower. So we try to make sure that we involve all, we can say, we needed to make sure that all the species that are within the gut for each patient are taken into consideration, so we can build this pairwise genome scale metabolic model for each healthy subject and the COVID-19 patients. Okay, so what we did, so as an output, we tried to extract those features that will be the feature for our machine learning pipeline. So we extracted the community growth rate, so that fluxes that I explained in the beginning. So we tried to extract the community one, and then we extracted the interaction ratio. What I mean by this is, imagine like we have different, we can say, interaction. What I mean by interaction, when two species, okay, are living with each other, okay, one can like come over the other. So for example, one can eat on the top of the other one and then make him having a low energy because he eat his like intakes, okay, he eat his nutrients, okay. And in this case, when someone like eats your meal, we call it a parasitism situation. And there is also other type of interaction. I will explain it in the upcoming slide, but there is other type of interaction that are either they are both there, they don't want to bother no one, and everyone eats with their own we can say nutrients, and this is a mutualism case scenario. And there is also other, we can say, type. So what we were trying to convey is like we were trying to have a sort of percentage or some sort of ratio to know how many type of interaction is within each patient. Okay, so the third feature in this case is the reaction exchange flux. So when we talk about the exchange flux, if you remember, I explained the transport reaction, those reactions that are communicating outside the cytosol. So it's those reactions that we focus on because it's there when it's very interesting to see how they communicate with the other species. Okay, and then basically, we tried mainly to use some unsupervised machine learning to understand how we can say, based on the community growth rate, all those features, if we can find a sort of two clusters, so two different cluster of the healthy subjects and the COVID-19 patient. Okay. After having the interspecies interaction, so those were fed to different classifier, and then we tried also an ensemble machine learning classifier to see if we can increase the accuracy and all the other metric, because we want to see how this will make sense if it's like used in a different case scenario. And then we tried also to get out some, we can say, sample level metabolic enrichment. So in this case scenario, we try to understand how we classify the sample based on the metabolic profile, and how we can, from those outputs that we can gain from the explainability, we wanted to give some biological interpretation and insights, okay? So if we zoom a little bit more on the framework, okay? So here is like a different panel, but here explain like the complexity of each panel. So starting from those two patients, we have those sequencing. As I explained to you in the beginning, we do a multi-sequencing to realize or construct the genome scale metabolic model, and then we make them communicate. We make like the GM refine, because we cannot just take it from a reconstructed tool like from CarveMe or a different tool that could easily give you the metabolic model, but we need to be sure that they are feasible, that they will give us some, they will quantify the growth rate. So the refinement take a little bit more time. So we were trying to have a high quality genome scale metabolic model. So second thing, if we want to zoom in, so we tried the two species community GM, and then basically from those two community GM, we took those interactions type, and then we added also some fluxomic data, such as those V1, V2 that I explained in the third slide, where I tried to explain what is the flux balance analysis. So all this we can say, the different, we can say, modality. So we have the exchange reaction, the growth rate, the interaction, all was integrated as a community feature. And then basically we end up by the model interpretation and the biological interpretation. Okay, so now let's a little bit dive in to the results section. Okay, so in the results section, here I mainly showcase in the first panel like the output of the mathematical model, the genome scale metabolic model. Okay, so we can see that we have an increase of number of pairwise GM, since we are making sure that all the species interact with all the bacteria within the gut. Okay, so in this case of scenario, I mainly will study the genome scale metabolic model. You remember to understand the quality of the genome scale metabolic model, I do a sort of score checking, okay? And here I check the consistency of the model, the annotation of the model. So if there is any problem regarding the annotation of the reaction, the metabolite itself, I need to be careful for the refinement. And when we talk about the gene annotation, it's not like very problematic case scenario, but sometimes some annotation are from a specific, we can say, database, like the Miriam database or the big database. And when you annotate based on a specific database, the score is less, because you use just this database to create your annotation from gene. So what I take into consideration the most is the consistency. So I mean by consistency is how my model will perform when I will do a medium composition, and how I will have like the let's say some basic biomass mass balance. If there is any mass balance problem, there is a problem, because I need to be sure that there is a mass balance. See, my stoichiometric matrix is give me feasible solution. So this is my main concern for the metabolic model. Okay, so after, if you see in the panel D, is like we're trying in this case of scenario to check each exchange reaction, if there is any statistical significance between each one, and if so, we want to see or compare those statistical metric with the metric that we will get from the SHAP value, okay? So we were trying to see the transport reaction, how they are different from the COVID case scenario and the healthy one. And then we were also interested in seeing the type of interaction. So the type of interaction that were occurred in our case was the amensalism, the competition, and the neutralism. So there wasn't any like interaction, and then either they are competing with each other, so both they are doing minus minus, so both they are negative interaction, or one is minus and one is zero neutral. So one is like mainly eating from the other one, okay? So, and finally, in the panel F, we were trying to understand like mainly how the community growth rate is help differentiate the COVID case of scenario and the healthy one, okay? So finally, this is like we can say the interval that we worked on for the machine learning classifier. So I just give like some parameters here. And these are mainly the outputs, basically based on the time, the precision, the recall, on the F1 score. So we're basically satisfied with the ensemble model, where we mainly achieved, let's say, a higher accuracy or similar to the gradient boost classifier. But in this case of scenario, we wanted mainly to see if the ensemble model outperformed other in terms of accuracy and precision and recall. So beyond the raw performance, we want to understand now why the model makes this specific type of prediction. So now I will move on like to the model interpretability. So in this case of scenario, we wanted also to check like for the different range. So using like the Shapley additive explanation, how we can quantify each feature. So I mean by this, the top contributor were the hydrogen, in this case scenario, the carbon dioxide, and the iron. So this feature mainly corresponds to biological meaningful pathway. So it's confirming that like the AI is not just the black box, but we wanted to capture here the real biochemical signal. So what I mean by this is I wanted to understand if those extracted features are making sense for my model. So, and in this case of scenario, mainly I try to study if, like, for example, based on the literature review, if like the small fluxes of the oxygen, or the mainly we can say the hydrogen intake, if there is any significant biologically in the key metabolic alteration that we have. So we try to observe that elevated CO2 and the hydrogen flux and the reducing of the iron are like mainly related to the COVID-19 patient. So this pattern mirror the clinical observation, and we can say the respiratory distress that a patient can like mainly have when we are dealing with the virus itself. So the respiratory distress here are mainly shown in this like all the exchange reaction that we focused on. And also we want to understand if we can suggest that the microbial metabolism may both reflect and influence the host respiratory function. So that's why we try to see the implication for the biological interpretation. So the iron is the central important component for the oxygen transportation. So we have our oxygen using like a sort of transporter, which is the iron, via the hemoglobin, and it's when we have a dysregulation, it's mainly linked to the hypoxia. So that's why sometimes when we try to understand the negative like value of the SHAP, and they're related to the iron oxygen, the hydrogen cycle, we can understand that this is mainly due to the hypoxia, and the excess of the dioxide of carbon, and in this case we have also the hydrogen, suggests also a sort of compensation of mechanism and the oxidation, the distress that the patient like have. So there is like some recent work in the literature that found that the hydrogen gas that sometimes they give to the patient during the healing stage can also mitigate inflammation. Sorry. So we wanted to understand also why, like, for example, the hydrogen is like also one of the important, we can say, exchange reaction in this case of scenario. So now, if we think of, so starting from the company that part funded my PhD 5 years ago, they worked mainly on the COVID-19 testing, so they are complement genomic. Starting from the COVID-19, we tried to explain the mechanic aware behavior that we have from the Pairwise GAM, and we try to understand the biological interpretation from all those, we can say, metabolic transport reaction, and how this influence like the inflammation disease itself based on the bacterial behavior. Okay, so the thing is, if we want to think beyond the COVID-19 as a case of scenario, we can use the same framework and apply it to other conditions, such as cancer, diabetes, or neurological disorders like Alzheimer, where we can understand how this gut and how like the changes that occur on the gut microbial can contribute to the disease. So it's very important because it's recently shown that this community, this complex behavior, are those one contributing to the disease. And it's important, as I said, to understand like this concept. So finally, just like to give you a small idea on how we can use this framework. So there is like a recent grant that we was like luckily accepted, which is a Horizon grant. So where we try to understand, or we try to unlock the industrial potential of the extramophilic microorganism. And here the idea is like using this community metabolic model to build those species, like sponge species, that are a little bit more complex than the bacteria, and how they interact in an extreme environment. Okay, so also, of course, we within like the I act as a small member on the school computing and digital technology, especially in women in engineering and STEAM projects. So here we are trying to celebrate all women achievements in the different careers, and especially in STEAM. And there is also other, we can say, implication to the model as well. So in conclusion, just to conclude, we can say that the Pairwise genome scale metabolic model can capture, we can say, those microbial interaction, and here we're talking about high quality resolution. And when we integrate those features that we take from the genome scale metabolic model itself, we can enable accurate classification. So the key finding is here to highlight some biomarkers such as iron, oxidants, and CO2 and hydrogen that could implicate some mechanistic link between the interaction itself and the disease state. So key finding of this small, we can say, example of how this framework can be used is like, as I said, the exchange fluxes using that is due to the transportation of oxygen, so the hypoxia. And then, of course, we can say at the end that the perspective of the model is beyond the COVID-19 application, which is like how we can basically use the multi-species on the top different omics, so the metagenomic, the metabolomic, the transcriptomic data, where we can achieve more accuracy because we can strain the model more, and then it's more specific into and give us more, we can say, help for personalized medicine and therapeutic prediction. I just finally a small acknowledgement before giving the room for some question. There is here my supervisors. We are very, we can say, I can describe it as a very good team because we all there helping each other. It's a very healthy environment. So everyone, I work on microbial, a lot of my colleagues work on cancer, and we are all try to merge our expertise and try to give like high quality work in the field. So yes, this is just my contact, and of course, if for the networking at the end, and thank you again, Redwan, and everyone in the community of MoroccoAI.
Thank you very much, Dr. Chaimaa Tarzi. It was very impressive. I really liked the presentation. I'm not an expert, I'm not very familiar with your field of application, but I learned a lot. I really learned, and I'm sure people who are doing AI for health in general, they will enjoy your presentation. So now we will move to the questions and answers part. We did receive many questions, so I have like yeah, 7 questions, which is nice. So I'll try to probably begin with more specific or maybe more broad questions and then move to more specific ones if you may.
Okay.
Okay, so the first question is, do you collaborate directly with medical doctors, and what are the challenges and difficulties
Mm.
you may
So,
face?
so the challenge is in the question is
Yeah.
when you collaborate basically with medical doctor is the data itself. So
Mhm,
we wanted this framework basically to be used for broader patient, broader we can say subjects. But the problem is the data itself. So there is like ongoing collaboration with the NHS, which is one of the national organization of health in the UK. But still, we can say the problem here is like the data, the availability of the data. There is images as well that are already there, but sometimes when we talk about the sequencing, sometimes we find like lack of the data
mhm,
availability in
mhm.
that sector. So we are open, of course, to like any we can say collaboration that will also bring us some data, and of course their expertise in the field, because I mainly think that the doctor or the biologist itself or the medical people will be more than happy to be within this we can say work, because they might see that there is a lot of correlation that you get when you interpret the model and you add explainability on the top.
Yeah, thanks a lot. So we do have another question, which is very, very interesting, I think. It's about well, the question is stated like this: how your work can be translated into the African context and the Moroccan context in particular. So this is the question. It's very general.
Yes, so how can we like use this in Africa or Moroccan case scenario is uh, take the diabetes disease is one of the disease that we have in most
Mm-hmm.
even like
Mm-hmm.
within our family, either one have a tension problem or diabetes problem.
Mm-hmm.
So if I choose you or anyone from Africa, they will have like this, we can say diseases that are within Africa due to a lot of reasons. Our diet, the way we eat recently, and everything. So mainly we need like there like a lot of volunteers, and we need like a clinician to be open and sign in some ethical clearance where we can mainly take some data from like some stool data to be able to capture the bacterial, we can say, relative abundance that are in each patient. And then there we can like start working on use the framework again and see like any, we can say, explainability of like a specific we can say metabolism that can interfere at the end on disease. So mainly Morocco already face growing need on the data driven biomedical research. So what I mean by this, if we use the same pipeline and we use it in a local health priority, we can achieve like very good indeed outputs. And then could be from stool, could be from the nasal or the saliva. So all those sample can give us a DNA sequence that I can start with, and then use my framework to understand the clinical outputs.
Correct. Thanks a lot. And this is, I mean, a question, but coming from me, do you know of researchers like in Morocco working on closely related subjects? Do you have any idea?
So uh, my original like diploma was on software engineer. So when
Hmm.
I did my PhD, I totally shifted in the biology field. So I'm a software engineer that I was thinking like maybe if I read a little bit on biology, I will it make more sense. I wanted like I love like designing like front end and back end web pages, but I was like, let's a little bit go out from my comfort zone and try to go to a field that I don't know anything about. My bachelor was I did preparatory classes in France, and my also my baccalaureate was a mathematics type B. So for those that are Moroccan, they know that when you do type B, you don't want anything to go with biology. So at the end, my PhD was about biology. So I still have a lot of contact with my professor in software engineering, that where I have like an opportunity, I share it with them. I keep in touch with them. But I would love to have like a sort of opportunity or opening to the microbial community and the people that even like work on pharmacy, because you can't imagine how this we can say framework can be applied, or other work that I do in the same time. This is just one of the work I'm doing. I'm also using deep learning for protein structure and everything. So you can't imagine how the system biology field is very broad, and I would love to have like collaboration in Africa, in Morocco, in this sector as well.
Great. Nice to hear. Yeah, let's go to the next question, since time, yeah, is starting to pressure us. So you mentioned gap filling and manual curation in your slide
Mhm.
from genome to model. Can you say more about it?
Yes, so if I go just like directly to the slide, so this is mainly a small, like, let's say, example. So sometimes when we talk about the gap filling, is like when we have a generated metabolic model, sometimes he generates that there is a reaction that is happened, but there is no metabolite being produced, or not metabolite to be consumed. So it's like is a or sometimes it generates a loop, like a reaction that produce and consume in the same time. So all this infeasibility that happen in the model, or all this like let's say missing reaction, problem on the production in terms of biomass, in terms of essential metabolite that been missing, it's happened. So because the tool itself, it's not perfect, so we need to gap fill using some biologically constraint, and we need to add those reactions that are supported by like some genomic evidence. So in this case of scenario, I did a work with a big lab in San Diego, where we wanted to gap. The paper was just about gap filling the Chinese hamster metabolic model. So there we tried to look on all the, we can say, those reactions that are not there, and we tried to see the evidence of each genomic, check the reaction stoichiometric matrix that I explained, and literally come back to literature and closely inspect if there is any problem regarding those reactions. So there is a lot of steps. I didn't want to put a lot of information in today talk, but it's another, we can say, open door that you need to work on, and you need to study to understand how you can polish the model, how you can make it, because in the beginning the model might give you infeasibility and might not predict or quantify those fluxes that I was describing.
Okay. I will take two last questions so that we leave some time for the breakout session. The first one is, so in your COVID-19 work, we see you use UMAP representations a lot. My remark is that it does not clearly separate the two classes. Do you know the reason?
Sometimes when we talk about the feature from the metabolic model, those fluxes, we can say values that we put all in as an input for the metabolic model, are a little bit near in terms of value. Okay, so the problem when we try to separate or do a sort of a clear separation of the UMAP, those features are not clearly saying how we can separate those, we can say, two classes. But since those metabolic features have sometimes just like slight difference, for example, when I talk about the exchange transport, the difference of in terms of we can say the millimole per gram dry hour are highly recognizable, but still biologically make sense. So that's why we have this problem when we are doing the unsupervised learning technique.
Okay. Okay, thanks a lot. Yeah, I did receive other questions, but sorry, I cannot take all the questions now, but please stay for the breakout session and you can ask Dr. Chaimaa directly. So the last question, metabolism swing with circadian rhythm, diet, and fasting state. Do you take into consideration these model temporal dynamics?
Yeah, as I said, like when we talk about sometimes like the metabolic model, as I said, we cannot not take into consideration the environmental, we can say, nutrients or any aspects. Sometimes we have some data about like how the model are changing in terms of time, so we can generate dynamic flux balance analysis. Sometimes we don't have, like in different time, we can say, scenario, which is the change of the model itself. But we always keep in mind, like in this case of scenario, we always keep in mind, we can say, the nutrients, so the constraint from the nutrients. So the first constraint, if for example we have limited glucose intake, or we have limited like specific metabolism based on the diet of the person, we need to put it as constraint for each metabolite. So sometimes we use a medium composition to make everyone have the same constraints. Sometimes we change those constraints based on the patient base. So what I mean by this, we can still keep the more complex way of the framework could be, instead of using flux balance analysis, we could use the dynamic flux balance analysis. And in this case of scenario, we need to take into consideration the uptake and the discrete of each metabolite in different time points. Okay, so it will be a little bit more complex. It's add more complexity layer, but it will help us, we can say, understand the dynamic of the microbe and how they change based on the time and how the flux change based on the time. But in terms of scalability, it will be a little bit more because we already have problem that each patient don't have the same microbes. So we needed to create a sort of community one so we don't have imbalance for our pipeline. So if we add the dynamic behavior, it's add another
It's more
layer of
complex.
complexity.
Very clear answer. Thanks a lot, Dr. Chaimaa. So yeah, we reached the end of our webinar. Dr. Tarzi, it was a pleasure having you with us. And yeah, for the people who are watching us on YouTube, I will now stop the recording and see you at the next webinar. And for the people who are with us on Zoom, please stay with us for a breakout session with Dr. Tarzi to ask you all your questions, especially those who I could not ask Chaimaa right now. Thanks a lot.
The pleasure is mine. The pleasure is mine. Thank you for inviting me. Yeah.
Our pleasure. So I'll just stop.